So welcome to the second session of the Frontiers of Science meeting. Today we have three very good presentations today focused on the human microbiome and in particular focusing on various diseases of the gastrointestinal tract. First up this morning we're going to have Dr. Claire O'Brien. So Claire received her PhD from the Australian National University at the Medical School in Canberra in 2012. And currently she is a NHMRC Peter Doherty Fellow and lecturer at ANU, and her main interests are in inflammatory bowel disease and in particular Crohn's disease. And her topic today will be presenting on the role of the gut bacteria in Crohn's disease. So over to you, Claire.
Thank you, Dave. And I'd just like to thank everyone involved in organising this meeting. I've thoroughly enjoyed the first sessions and last night, and I'm sure there's more to come. So there's two major forms of inflammatory bowel disease: ulcerative colitis, which affects the large bowel only, and inflammation is superficial, and then Crohn's disease, which can affect any part of the gastrointestinal tract from the tongue right through to the bum. So inflammation in Crohn's disease is transmural, meaning it affects the full thickness of the bowel wall, and the terminal ileum, where the small bowel meets the large bowel, is most often involved. So genome-wide association studies have identified over 200 genetic variants associated with the IBDs, but genetic factors alone can't explain a number of key observations. For example, the prevalence of these diseases are rising faster than population turnover. The penetrance of the disease variants is very low, so there's a lot of carriers out there who don't develop disease. And concordance between in monozygotic twins is only around 58%. So we currently think that the IBDs result from an abnormal ongoing immune response in response to environmental factors, which are likely luminal bacteria in a genetically susceptible host. So there's around 33 Crohn's disease specific gene variants, and the most significant replicable mutations encode genes involved in bacterial sensing, so NOD2, autophagy, which is intracellular killing of bacteria, and signalling in response to bacteria. But there's several other lines of evidence to suggest that microbes play a role. For example, aphthous ulcers, and there's one shown here on the slide, overlie Peyer's patches in the small bowel or lymphoid follicles in the large bowel, and these are lymphoid aggregates and they're the site of luminal antigen sampling. Crohn's disease lesions also preferentially occur in gut regions with the highest bacterial concentrations, so the terminal ileum and the colon. Diversion of the fecal stream reduces pathology in downstream mucosal sites, and it's only once you reinstate the fecal flow that pathology returns, and most animal models require bacteria for the onset of colitis. So before I talk to you about what goes wrong with the microbiome in Crohn's disease, I just wanted to draw your attention to this study by Chris Huttenhauer et al. That was published a few years ago in Nature. And what they did was they looked at the microbiome of over 240 healthy individuals, and they did this using 16S gene sequencing as well as shotgun metagenomics to look at the function. And so in the top graph you can see each vertical bar represents the microbiome of a single patient, and it's at the phylum level, and there's huge diversity in the microbiome of healthy individuals. So the ratio of Bacteroides and Firmicutes differs quite a lot within the healthy population, but down here you can see that the function of the microbiome for the same microbiome above is consistent across healthy individuals. So a core function of the microbiome is obviously carbohydrate metabolism, and it's consistent, it's the green bar across the top there. It's consistent across those microbiomes. So harping back to what the panellists were talking about before, function's probably more important than who's there. So in Crohn's disease, we see decreased alpha diversity, or the number of different species of bacteria, and a reduction in some of the core Firmicutes bacteria, such as Faecalibacterium prausnitzii, which produces an anti-inflammatory protein called MAM, which apparently exerts its effect through the NF-kappa B inflammatory pathway. And then you see this concurrent increase in pathobionts such as E. coli. And this results in a reduction in butyrate and short-chain fatty acid production. And butyrate is the major energy source of our colonocytes, or epithelial cells in our colon, so butyrate's very important for gut health. We also see a reduction in amino acid synthesis by bacteria. And then we see traits that are more characteristic of pathobionts or pathogenic bacteria. So we see an increase in auxotrophy, an increased propensity for bacteria to tolerate oxidative stress, and utilisation of the type 2 secretion system. So studies in our lab have aimed to identify putative bacterial triggers, and the list is long in Crohn's disease. We've also sought to look at functional triggers such as variants of Fimbriae within E. coli, look at bacterial community imbalance. I don't like to use the word dysbiosis, but people usually know what you mean when you say the word, but alterations in the community structure, and more recently, host microbe interactions. And to do this, we've got access to a number of different tissues. I work very closely with gastroenterologists, and we have early stage samples. So when I talk about early stage disease, I'm not talking about new onset, I'm talking about those aphthous ulcers, which I showed you a picture of. And they're the same thing as the ulcers that you get on your tongue, but they just develop into larger transverse linear ulcers and develop into eventually late stage tissues, which are the bowel resections. So when things go horribly wrong and patients need to have part of their bowel removed. So to date, we haven't found evidence to support the role of many of the putative pathogens, in particular Mycobacterium avium, subspecies paratuberculosis, or MAP. The only one that we've ever sort of found any evidence to support a role in Crohn's disease for is Escherichia, or at the genus level. But in early stage Crohn's disease, it's present at about the same frequency as healthy controls. In late stage disease, it's a different story, and you see an increase in Crohn's disease relative to non-IBD, and this figure's even higher if you look at just terminal ileal tissues. So we've pursued the E. coli sort of story, and there is a pathovirus of E. coli associated with Crohn's disease, referred to as adherent invasive E. coli. And it's more frequently isolated from mucosal biopsies of Crohn's disease patients than controls. And to be deemed AIEC, the strains have to be able to adhere and invade intestinal epithelial cells, as well as survive and replicate within macrophages. And this is the only way to determine whether a strain is AIEC, and as you can imagine, it's very laborious doing these phenotypic assays. And you can see an AIEC strain that survived and replicated within a phagolysosome of a macrophage here. So we conducted a study of 42 B2 phylogroup E. coli strains, and we determined the AIEC phenotype for these strains. And in this tree, you don't need to be able to read the labels, you just need to know that the strains in red are AIEC. And you can see that they're spread throughout the phylogenetic B2 E. coli tree. So they're a very heterogeneous group of E. coli, and they in fact occur across all major groups of E. coli. And we were unable to identify a gene or base composition differences that were unique to these strains. So they're more akin to your UTI isolates, where they're more likely to, say, be a sequence type 131 or something, but you can't actually define them by a single gene or base composition difference. So sort of shifting gears a bit here now, and sorry for those of you who are a bit squeamish, but this picture illustrates the complications that occur in Crohn's disease. This is a classic Crohn's disease stricture, and I like to show it because Crohn's disease is a hidden disease. You know, you wouldn't know if a person sitting next to you had Crohn's disease or not, but this is the sort of thing that's going on inside them. So anyhow, we did a study where we sampled involved mucosa, so obviously where the stricture is there, the scarring that's built up and hasn't allowed food to get through, as well as uninvolved mucosa on the side here. So these normal folds over here represent normal bowel. And then lymph nodes, which are on the outside of the bowel walls, so the mesenteric lymph nodes. And the reason we did this was because we thought bacteria might translocate the gut, and then immune cells would selectively take up the trigger, drain to the mesenteric lymph nodes, and we'd have fewer bacteria to look at there than in the gut where there's hundreds of species. But alas, what we found was that there was non-specific translocation, and what's in the lymph nodes is the same as what's in the involved mucosa and uninvolved mucosa. We did, however, find that there was reduced diversity, which is a common finding in Crohn's disease, and really stark community imbalance. You know, patients presenting with sort of 90% proteobacteria. So we've turned our attention to the early stage disease tissues now, the aphthous ulcers. And interestingly, we found that there's no community imbalance in these samples relative to healthy controls, but reduced diversity is a feature still of the early lesion. We don't see a reduction in Faecalibacterium prausnitzii, which again is quite interesting, and this is probably the first study to show that it's not reduced, but it's also the first study of this earliest lesion because they're really hard to get, and these patients have also been, haven't been on medications either. So in summary, the gut microbiome is altered in Crohn's disease. It's characterised by decreased diversity, an increase in pathobionts. We see abnormal community structure in late-stage tissues, but not early-stage tissues. We don't identify common organisms in lymph nodes or mucosa, but E. coli is still a possible trigger. So in a current study, we've been culturing E. coli from the lymph nodes of patients, and we've been able to do this for 60% of patients, but haven't been able to culture E. coli from a single control lymph node yet, so that's looking promising. We've got a PhD student working on characterising the viable communities using cDNA, as well as temporal changes in patients, in the same patient over time. And I'm also looking at host microbe interactions, so looking at host gene expression and then marrying it up with the bacterial communities and the virome as well. So I'd like to thank my major collaborators, as well as past students, study participants, and all the organisations that have funded my first few years of postdoctoral research. Thank you.
So our third and final speaker for this session hopefully won't have too many gastrointestinal tract pictures for us prior to lunch. So next up we have Dr. Erin Shanahan, who obtained her PhD from the University of Sydney in molecular microbiology and host pathogen interactions, and currently works on the role of the microbiome in gastrointestinal disorders and facilitating collaborations between two groups at the TRI, the gastrointestinal group and the metagenomics group. And currently, Erin is researching the microbiome in gastrointestinal disorders, and today presenting life in the small intestine tract, the forgotten microbiome.
Hi everyone, I'd just like to say thank you for the opportunity to talk to everyone today, and thanks to the organisers for what's a great meeting so far. So. As mentioned, I'll be talking about the small intestinal microbiota. So just to jump straight in, why are we interested in the small intestine? So there's an enormous number of studies on the GI microbiome, but what people may or may not realise is that the majority of these have been done on faecal samples. And this doesn't necessarily represent, or certainly doesn't represent, what's going on in the entirety of the gut. So it's important to think about all of the different regions of the gut. So the small intestine, it's got the largest surface area in the gut. It's also got a thinner, more permeable mucus layer compared to, say, the large bowel, and this is to allow for digestion and absorption to occur, but it also creates the potential for increased interactions between microbes and the host. It's the site of induction of many symptoms that patients may experience, so for example in irritable bowel syndrome. It's a highly vascularised site, so lots of blood vessels, and this is important for, say, the gut-liver axis or some of these gut metabolic axes that we see. And it's also highly innervated, so that means it's got lots of nerve endings. And this is really important when we think about gut-brain axis. And as I mentioned, a key site of digestion and absorption. So it's an important region. Historically, particularly the first part of the small intestine has actually been considered sterile, similar to the concept about the stomach being sterile. And it's also very poorly characterised compared to other regions of the GI tract, and a lot of this is to do with the difficulties in sampling this region. We also have concepts of, and this is linking to the sterile nature of the environment, or potentially sterile, of small intestinal bacterial overgrowth, where we don't actually want too much bacteria present here. But this is very difficult to detect and is very poorly characterised, and in clinical practice leads to, well, as a microbiologist, what we would consider injudicious use of antibiotics. So there's a few issues going on here, and a lot of it's to do with it being a poorly characterised region. So I've said the small intestine, but it's actually a series of different niches. So as a microbe, what would I experience if I was in these different niches? So the first part of the small intestine, the upper portion, we're talking about the duodenum and the jejunum. So this is a very harsh environment. It's characterised by the presence of acid, chyme, that enters from the stomach. You've got digestive enzymes, bile, influxes of food and nutrients which then move through, and quite a fast transit time. As you start to move lower in the tract, you move to a more favourable environment for microbes. Things start to slow down, you've got more fermentation happening, a more neutral pH, and this proceeds as you get to the end of the small intestine, which is this terminal ileum, which is more similar to the large bowel. So it's a series of different niches as you move along the tract. So I just want to highlight the importance of the mucosa-associated microbiota, and this is particularly relevant for the small intestine where you have this motility. So this results in the low load in particularly the first part of this organ, and it also results in transient organisms, so things that are moving through, and the luminal contents in particular are moving through. Therefore, the mucosa-associated microbiota, so what's adherent to the mucosa, is more likely to be more permanent and is probably going to be quite important in terms of mediating effects in this region with host interactions. Okay, so how do we actually sample the small intestinal microbiota? So our samples are biopsies, and they come from patients who are undergoing endoscopic procedures. So this is either an endoscopy through the mouth to the first part of the small intestine, the duodenum, or a colonoscopy, which is going up the other way, and we can reach the last part of the small intestine, which is the terminal ileum. So we obtain biopsies in this way, and these are the samples that we work with. We've, as I said, got to focus on the mucosal microbiota. Our group has also tried to develop sampling techniques that try to avoid oral and luminal contamination when we're taking these biopsies. So this is through the use of particular biopsy forceps, which are sheathed and protected until you take your actual sample. And also focusing on DNA extractions and preparation methods that are optimised for low bacterial load samples. When we get these biopsy tissues, we end up with greater than 99% human DNA. So you've really got to think about your methods to accurately detect the bacteria. And no one wants to be characterising their reagent microbiome. So having worked through some of these methods, I just wanted to briefly touch on the composition of the different parts of the small intestine. So this is some data looking at a couple of these different regions. So if we compare the duodenal and gastric microbiome, there's actually a reasonable overlap between these. We can't really clearly distinguish the populations. On the other hand, if we compare duodenal and terminal ileal microbiota, you can see that there's a striking difference. The duodenal samples are quite similar to each other, and I think this reflects the fact that this is a very harsh environment. It's it's very selective, whereas with the terminal ileal samples, there's a much greater spread, so greater into individual differences. And it's a bit hard to see, but we've got a couple of rectal mucosal samples here which sit in the middle of the TI data points, and this indicates how similar the TI becomes to the large bowel. So what's actually there? Looking at the duodenal biopsy samples, we see an overwhelming dominance of Streptococcus, and then lesser numbers of anyone who's familiar with the oral microbiome would note that these are very similar to the oral microbiome, so Veillonellas, Fusobacterium, Actinomyces, Prevotella. So looks like the oral microbiome. On the other hand, the terminal ileal microbiota is much more like the large bowel. So you're starting to see your typical Rhiminococcaceae, Faecalibacterium, Bacteroides, etc. So as I said, we say the small intestine, but it's actually either a series of niches or a gradient as you move through. Okay, so that was a bit in general about this area, but what about how this relates to particular diseases? So one of the things that we're interested in our group are the so-called functional GI disorders. And these are disorders that are classified based on symptoms. They don't have an obvious organic cause or obvious inflammation, say like Crohn's disease does. And the typical examples are irritable bowel syndrome and functional dyspepsia, which is a more upper GI type symptom. So these are greatly increasing in prevalence. And one of the big issues with them is that the treatments target symptoms, so we're not actually getting rid of the underlying cause. And also diagnosis is very expensive. So you have to you have to rule out overlapping diseases such as reflux disease, which have similar symptoms, but obviously have a different cause. And I think one thing that really stands out to me is that only about 25% of patients who come in to have an endoscopy procedure actually have a cause found for their symptoms and are put on a treatment. So this is an incredibly expensive, procedure to then not have an outcome really for 75% of people who have it. So we need to actually start to understand a bit better about what causes these disorders, and the understanding that it's not just in people's heads that they have an upset stomach. So one of the things that's come out recently is that there's actually low-level inflammation in these functional disorders. What we've noticed, so this is focusing on the upper GI functional dyspepsia syndrome, and we can see that there's infiltration of eosinophils or mast cells into the duodenal tissue. There's also an increase in gut-homing T lymphocytes, and these cells produce greater amounts of cytokine, and this, importantly, is actually associated with symptom intensity in these patients. So this is not overt inflammation that you can see during an endoscopic procedure, but it's a low level of inflammation that is there ticking over. So obviously, what is driving this? And the microbiota is a key potential driver. So I just outline the project that we're undertaking to try and investigate some of these questions. So we have a large-scale study running in which we're recruiting dyspeptic patients, IBS patients, and control subjects. And in all of these subjects, we obtain esophageal, gastric, and duodenal biopsies. These are for microbiome, RNA, and histology analysis. We also get additional duodenal biopsies, which we're using for both immune cell isolation and characterization, and also bacterial culture. We get blood from the patients so that we can phenotype the peripheral blood or immune cells. And the patients also undergo full clinical workup and a lot of functional or clinical testing looking at their responses to meals, response to nutrients, their small intestinal permeability, and these type of things. So this links back to what was mentioned this morning about how important it is to have excellent metadata if you're going to really dig down to what's really happening. One thing I would point out is that our immunologists think that the microbiome data is their metadata, and the clinicians think that all of our lab data is their metadata. But I think that highlights the fact that it is multidisciplinary, and if you're going to do it well, you need to have the people who are good at those different aspects of the project. So that's an ongoing project. We're still recruiting patients, we're still analysing all of that enormous number of samples and data, but I just present some pilot data that we have from the study. And this is looking at the duodenal biopsies from these patients and looking at the microbiota. So this is looking at the genus level at some of the bacteria that we observe in these biopsies. And what we found was that in the dyspeptic patients, there's actually an increase in the abundance of streptococcus, and then this is linked to then a reduction in some of the anaerobic organisms that we see. And we can certainly see a differential clustering of the two cohorts, controls and our dyspeptic patients, indicating that there is an overall difference in their microbial communities. And also importantly, we start to look at the microbiota and the link to the symptoms that the patients actually experience. So here I've just presented a couple of symptoms or clinical markers that we have. So one is patient-reported quality of life, and one is the symptom response after a meal. And what we found is that as diversity increases, we have a higher reported quality of life. As diversity increases, we have reduced reported symptoms. And then just looking at total bacterial load, so this is not particular bacteria, but overall the amount that we detect, we see a trend where we have increased bacterial load and reduced reported quality of life, increased bacterial load and increased symptom responses. So this is, I think, really interesting. We're starting to actually link some of these factors to the symptoms that patients experience. And we're in the process of doing this in a much larger cohort. So just to quickly touch on another disorder that we're looking at, and that's chronic liver disease. So this is a highly prevalent disorder. It can have infectious causes, hepatitis B and C, or lifestyle type causes such as non-alcoholic fatty liver disease. This is an emerging major burden to the health system with the increasing metabolic disease in the population. And what happens is that the chronic inflammation in the liver can lead to fibrosis and cancer, but not all patients will go on to develop cancer. And we're trying to understand why this is. And there's emerging evidence that translocation of bacterial products from a leaky gut can actually promote liver inflammation. So like we heard earlier about leaky gut and visceral fat and inflammation, the liver is also an important factor here. So similarly, we looked at duodenal biopsies in this particular data set. Again, we actually saw an increase in the streptococcus that we detected in the community. And we can also observe differences in the overall microbial community between our controls and liver disease patients, and as is reported in many disorders, a decrease in diversity of the microbiota. When we stratify this, depending on the underlying cause of the liver disease, we see that a lot of that, or the most obvious changes are in patients who have a hepatitis C-driven disease, and we're in the process of trying to understand why this might be. Something interesting that came out of this study was when we looked at intestinal permeability, and we actually see that the HCV, so the hepatitis C patients, have the greatest increase in gut permeability, so a more leaky gut compared to controls. And so this is quite interesting in this context. But all of this, I guess, raises some typical chicken and egg questions. So what's causing what, does the dysbiosis come first or second? And so just finish off by talking about how we're starting to try and answer some of these questions. So, and to make the point that we've got all this sequence data, but does it actually represent live bacteria that are doing something relevant? So, therefore, we've been culturing from our mucosal biopsies and then identifying and characterising any isolates that we get out. So this is just a partial list of the panel of isolates that we've been able to get out from our duodenal biopsies, which is quite and there's more than this now, but quite a number for a sterile area. And we identify these using various molecular techniques. And just to point out that we're starting to see a good overlap between what we detect in our sequencing and what we can actually isolate, which is nice, obviously. So what does this allow us to do? It can allow us to do culture-based bacterial load, so live bacterial load. We can much better functionally characterise these isolates. We can do more detailed metagenomics or metatranscriptomics. It's technically challenging to do this straight from the biopsy samples when there's so much human DNA present, so this gives us some other options. And looking at other metabolites, volatile organic compounds and this type of thing in more detail. So just very briefly, some very basic functional testing that we've been doing, one of which is, can the bugs actually survive the different pHs that we see in the gut? So as you know, the stomach is very low pH, especially during fasting. Duodenum is much higher, sits at about 6. After a meal, both the stomach and the duodenum sit around pH 5 before gradually moving back to their fasting state. So we test some of our isolates to look at how well do they survive in these different conditions, and we can see that yes, obviously they can survive quite well in the duodenal niche. And this is an important factor also because of the incredibly high use of proton pump inhibitors in many patients. Now these act to decrease acid production in the stomach, and there's, it's an important issue if you're reducing that acid barrier, what's that doing to the microbiota? So that's one of the things that we're also looking at. And so just finally how can we possibly link this to symptom generation in some of these patients? And what we do is co-incubate our bacterial isolates with different cells. When we look at epithelial cells, so like the gut lining cells, the isolates are, to be honest, not doing that much. But then when we look at immune cells, so this is cells, peripheral blood, mononuclear cells, we do start to see a little bit greater cytokine production, which is not surprising, but it's very important to think about in the context of increased gut permeability. In our patients, we see an increase in our gut homing cells. And so we can start to link and look at whether particular isolates are more likely to be driving that inflammation. So just to summarise, the small intestinal region is, particularly the first part, is a low bacterial load region, but there are niche-specific microbiota throughout the tract. We can see differences in this microbiota in different disease states, and both bacterial composition but also bacterial load overall may be an important factor. And obviously we've then been characterising these bacteria because we have the bacterial isolates. So just to finally say, I think that the sequencing data is a great way of trying to identify some of the different microbes and what they might be doing, but we really need to, as previous speakers have said, move on to more functional characterisations, and there's various methods of doing that, but one of the ones that we're focused on is starting to actually culture the organisms and go back to some of those more, the microbiology basics, I guess you would say. So that just leaves me to thank everyone who's been involved in the study and our funding, so thank you very much.
Thank you very much for that talk. I'd like to now call up our other two panellists. If you want to come up and take a seat, we'll have a discussion. So we have around about 30 minutes now to have an open discussion. So anyone who has questions for any of the panellists, you're most welcome to get the attention of one of the roving microphones and direct it to one or all of the panellists. So who's first with a question?
One for Claire. You mentioned that you were isolating E. coli from the lymph nodes. Are you doing any strain typing or characterisation on those isolates?
Yeah, when we get enough isolates, we'll be doing whole genome sequencing and comparing the genomes of those particular isolates. So, yeah, very soon we're doing that. hopefully we'll come up with something. You know, hopefully they've got something in common. I'm sick of all these negative findings and not getting anywhere. But yeah.
So I'm Simon from University of Newcastle. I guess this is directed at Claire initially, but I think it's probably relevant to the panel as a whole. So diseases aren't generally static. They have a natural course or a natural history, and say Crohn's disease very early on is a very inflammatory disease. But you've shown a stricture there, which is actually a far more fibrotic disease, and that's much later on in the course of Crohn's, and there's less inflammation involved. So I'm just wondering, has anyone or have you looked at whether the microbiome changes from the early inflammatory type to the later fibrotic stricturing, penetrative Crohn's disease? And I guess, you know, again, with functional GI disease, it would be the same type of question. You know, if you have, say, a diarrheal, chronic relapse of diarrhea or constipation, is the microbiome at that snapshot different, and does it make a difference to, say, disease progression?
Yeah, it's something that we're trying to address at the moment. We've been collecting biopsies and resections since 2007 when I started my PhD. And so a PhD student is now looking at patients over time. And so we have different samples from different patients. They might be biopsies, they might be parts of their resections stored away. So we're currently trying to address that and see what the course is, as well as look at the host gene expression at the same time to see, which will address whether, you know, what the levels of inflammation are at those different time points. But it's tricky getting the samples, but we're embarking on it now.
Excellent. Hi, my name's Sue Egan from University of New South Wales. I have a question that's about pretty much just general to everybody and maybe tied in semantics, but I'd like to actually understand a little bit more about the terminology of pathobiont and how, if anyone's doing any work actually in what role they might have in a healthy host. Does that make sense? Because it seems to be the terms obviously being used, correct me if I'm wrong, to explain sort of like an opportunistic pathogen that has a chronic sort of effect, right? A chronic disease apart from, as opposed to the acute. Do people actually look at these? Obviously things like E. coli are commonly found there, so where do you draw the line? Do you understand? It's, again, more semantics, more philosophy, but...
Yeah, I mean, I think it's a good question. I think that, you know, it's clear that, these organisms they're present in the gut in healthy individuals, and why is it that they then become associated with different types of disorders or Crohn's or anything else? I think one of the important things is, this is a microbial community, and there's all sorts of interactions occurring between different organisms. And so, is it actually the other organisms that are helping to keep it in check? And when those change, that's when this one is more suited to a particular environment that becomes present in the gut. So whether it's inflammation, poor oxygen levels, and it is able to then, better survive. And it's not even necessarily that it has a more intrinsic ability to cause the disease, but it's got an ability to survive those particular conditions.
And I guess that comes back to what Lewis was saying last night. There's just bacteria, not necessarily good and bad, and that intrinsic relationship from one bacteria to the other is also very important, where they have a synergistic effect. I think we have one over here.
Hello. I'm Mark from the University of Sydney, and I'm not a biologist, which is why I ask silly questions, but I've heard this term dysbiosis used quite a lot, and it strikes me that that's quite a hard thing to nail down, because what's functionally healthy might change from one individual to the next, and there could be many different kinds of dysbioses, maybe even of different kinds of diseases. How do you reconcile all of this? Can you spot a dysbiotic community just by looking at the community, or do you actually have to look at whether or not there's mass inflammation in the host?
Well, I guess the broad definition of dysbiosis is we compare the microbiota in healthy individuals and diseased individuals, and when we see they are different, then we say, okay, there's dysbiosis in the diseased one. We don't actually have a hard definition of what is a bad microbiome. Obviously, it is now clear that just identifying the species are not enough. We really have to look into the functional aspects of what the microbiota collectively, what do they do. I don't think we can define what is a healthy microbiome and what is a bad microbiome for now.
Yeah, I think that's really true. And I think, you use dysbiosis as an umbrella term for something looks different, but we don't really necessarily know what it is, but you need a word for it, I guess. But I think also something that's important is thinking about what some of the earlier speakers today mentioned, which is all of these insults on our microbial community. And we say we compare to healthy controls, but how healthy are they really? And is that a normal microbiome? So I think dysbiosis is a convenient word to use when we don't know the exact answers, essentially.
Yeah. It's a term I don't particularly like either. There's no, like they've just said, there's no definition, there's no uniform way of assessing it. Is it a 30% shift in phyla one way or the other? People have come up with their own ways to sort of calculate it, but it's not a consistent term, and some people even include diversity in their meaning. So I prefer to refer to the community structure or the diversity separately, and so people sort of understand what you're talking about a bit better.
We would also almost need to define healthy as well. What is healthy? There could be underlying diseases. The microbiome changes throughout age, so then you've got to age stratify everything as well. Very difficult question.
Yeah, it's incredibly complicated. And I mean, when we look at our biopsy samples, is a truly healthy person coming in to have an endoscopic procedure? No. So we use iron deficiency anemia screening patients who are found to have normal mucosa and no mucosal disease, but they still have iron deficiency, and obviously iron is incredibly important for microbes. So it's, what is a healthy microbiome? It's incredibly difficult to try and characterize. And then, all the insults that come through antibiotics and medications and diet, and it's, very difficult.
Britt?
Hi, I really enjoyed the talk by Jan about gut permeability, and I was quite surprised with the effects of fish oil. The animals that you used were fed ad libitum, yeah?
Yes.
So I'd just like to share something that we had done in our lab. I do a lot of human health, but I also do a lot of agriculture. And we did one experiment with chickens where we just a fitsidextran trial, where we had birds fed ad libitum and birds that were given 1 meal a day or 2 meals a day. And then we stopped feeding them. And we found interesting thing that birds that were fed ad libitum, that were used to have food and nibble all the time, within 3 hours their gut became leaky and it just started leaking. So they could not cope with hunger at all. We did not proceed with this, like we didn't continue to look at the reasons or stuff, but it kind of opens another story about it's not just about diet, it's about also how you eat that food.
Yeah, I guess it goes along with all the recent research on intermittent fasting or alternative day fasting. I guess there are some literature in animals they've been put on alternative day fasting. So one day they were fed, they were allowed to eat as much as they want, and the next day they couldn't eat anything. So in the end, if you average the total food and calorie intake, they were exactly the same. So on the day that they were allowed to eat, they will eat twice as much. So the next day they are okay with not eating anything. But there are some research looking at the microbiotas in these animals, and they do see some very drastic changes on the fat day or the fasting day. So obviously, the frequency of food intake and how much they eat really matters in terms of the microbiota changes.
I'm down here.
So as you saw by my talk, I'm definitely somebody who looks at the oral microbiome, and so I was quite surprised to see such an oral signal in the small intestine early on. But that really got me thinking about how there's very different communities as you move through the gastrointestinal tract. And what do you think the major factors are that drive those very different bacterial communities in such a small area? Certainly it's interesting to know that those oral bugs don't get any farther than kind of the small intestine. And so is it simply bacterial competition, or is it the physiology of the host and the ability to bind onto epithelial cells or acid, or what do you think that these factors are that drive these very different communities in the gut?
Yeah, I mean, I think the physiology of the host is probably a major one. When you think about the different regions of the GI tract, you have changes in motility, so how fast things go through. You have changes in oxygen tension, so the large bowel is very anaerobic compared to higher up, and that changes the ability of which organisms can survive. You have different levels of mucin, which are less higher up a lot in the large bowel. Any number of factors like that, I think, are going to be acting as selective pressure on microbes and determining which ones can survive. But I think also, as you start to get usage of what we eat by microorganisms, that also then starts to create an environment that's suitable for other organisms. And so some of those fermentation processes, as they start to occur in the gut, also then determines what the conditions that are more favourable for different organisms. One of the other things that we're also interested in is, I guess, sort of adherence and colonisation. So in the oral cavity, this is quite well established about these sort of pioneer species which then allow biofilms to form, for example. And so looking at how that may also work on the intestinal mucosa as well.
Have got any more questions? No? Okay, so we have 20 more minutes of question time if there are any, otherwise... Porrick at the back.
Hi, Porrick from the University of Queensland. So looking at the presentations, and those three more recent ones on the other presentation, it strikes me that we're still really at the level of associations and correlations. So I guess my question to the panel as a whole is, how close are we to being able to move towards causation? So to being able to link specific genes with disease course. That strikes me as being the challenge really at the moment.
I think with Crohn's disease we're far off. I think there'll be huge changes once the cost of whole genome sequencing for the host genome plummets, because in Crohn's disease, as I said, there's 200 genetic loci and patients can have any suite of those genetic variants. So to assess, defects in autophagy and how bacteria may be implicated in that particular scenario will be different from mutations in, say, sensing bacteria. So I think we've got a long way to go for Crohn's disease, but we're taking dolly steps all the time.
So I would answer the question by saying it's probably also different for different disorders and different sites of the intestine. So areas that are better characterised in terms of composition are probably now further along the pathway of functionality and trying to link that function to induction of disease. The poorly characterised regions and the poorly characterised disease as well. If we haven't even done the composition, then you haven't even started down the pathway. So I think there is still a role for some of that, but probably in other or certain disease contexts, it is time to stop just sequencing everything and hoping for some correlations and start doing some more mechanistic type studies.
Claire mentioned that is a long way to go for Crohn's disease. I think it's even further away in terms of metabolic diseases. I guess a good example is with patients with Crohn's disease. So we know that they have a leaky gut. We know that they have an inflamed mesenteric fat, so it ticks all these boxes. But interestingly, these patients with Crohn's disease, they don't normally have insulin resistance or metabolic dysfunctions. So clearly, this suggests that gut dysfunction per se is not sufficient to induce metabolic diseases. So in terms of metabolic diseases, we do need a combination of everything. Gut is one of them, but not the only one of them. So I guess in our field, our feeling is like what Lewis mentioned in the plenary yesterday. Probiotics are not going to fix everything, and it will not fix everything. But we do see probiotics as has a great potential in terms of preventing the onset of metabolic diseases rather than a one-off treatment.
So just to add to that, I think we've done quite a good job in infectious diseases in understanding what are the particular bacterial products that drive the disease, drive inflammation. How do host genetics influence that as well? So we probably need to start thinking about some of the techniques that are used in infectious diseases. Let's apply those to what we're looking at in the microbiome. And not that the bugs in the gut or wherever are pathogens or infectious organisms, but we can utilise those techniques to try and answer the questions that we're trying to answer.
And I guess the other thing too is if we know that the microbiome is a possible cause to some of these diseases, do we need to stop looking at what the bugs are actually in the gut and trying to find solutions as to their effect? So leaky gut, can we find something that can actually aid in preventing leaky gut, which leads into these diseases? So for myself personally, I investigated fatty liver disease and found that there was this increased intestinal permeability. I used probiotics and we had a reduction. So I never went and looked at the microbiome per se, but there had to be some effect there on the microbiome that prevented this leaky gut. So there might be something for future collaborations, trying to get the microbiologists together with these people who are investigating these chronic diseases, join the two together, and we might be able to get a solution.
You could argue that it's driven by resistant starch and fibre intake. That's a lot of the work that we do at CSIRO is understanding how upping your fibre intake produces more butyrate, and that then obviously tightens up the junctions in the gut. And so that seems to me, diet seems to be a much overlooked component of a lot of these studies in disease. So
the question is, if we increase these tight junction proteins, then we reduce the leaky gut. Does it then matter what the bacteria within the gut is if they're not able to get out to cause the inflammation as well?
Yeah, I think those studies have a lot of potential for some diseases or abnormalities of the gut. With Crohn's disease, we know that none of the genetic variants encode genes involved in the epithelium, so we think it's sort of an inside-out sort of disease. So I don't think diet approaches are unfortunately going to help us with Crohn's disease, but yeah, it would be nice if it could, if these sorts of approaches could improve symptoms, then that'd be great too. Yeah.
Tony?
I think the interesting thing about Crohn's disease, we've got various segments in the bowel that are displaying the pathology. I suggest that it might have nothing to do whatsoever with the microbiome, but what it's related to is the particular gene structure of that tissue. and it could be rewarding to do gene sequence studies on those areas that are affected by comparison with adjacent nearby areas. And it may be necessary also to consider the RNAs, because the RNAs now obviously play a major role. But it may be that's the answer, and forget about the microbiome. Of course, it would influence maybe the bugs that are cultured there, but it's really the intestine itself speaking to them rather than the reverse.
Yep. Well, luckily we're doing that right now. So we're looking at host RNA-seq data for unmedicated patients, looking at the aphthous ulcer, the first lesion, as well as adjacent unaffected mucosa. And then we've got healthy Pay's patches from normal controls and adjacent normal mucosa. And there's a lot of stories to be told. Yeah, this is whole transcriptome, host transcriptome data. Yeah, so we see a lot of genes upregulated in response to bacteria, inflammation, and I shouldn't give too much away, but there's a lot going on in terms of the host responding to bacteria.
You're saying you're seeing it in relation to the microbial infestation. I'm saying it's the tissue that's determining.
Yeah, so the RNA-seq data that we have is for the host. It's the host tissue, not the microbes. We've looked at the virome and the bacterial communities for the tissues, but we now have host gene expression data for those biopsies, which is telling us a lot between the host microbe interactions. It's there's another challenging data set.
Any more questions? Otherwise, I think we'll have a lunch break now. So just to reiterate what's been told this morning, the posters are still up around the corner there. So please, throughout the lunch break, go around, interact with the posters there, and ask any questions of the people who have the posters. And I think we're back at about 1 o'clock. 10 past. Okay, thank you.